Good.
Great. So my pleasure today is to introduce Vivian Polkinghorn. Vivian comes with us from the Madison area. She is a biochemistry major who's kind of interested in the pharmaceutical industry. And she and I have been working for some time, at least a term, really trying to tease out some of the both molecular scale and kind of, I guess, epidemiology scale of thinking about how antioxidants and chemotherapy work together or don't. So with that, I'll turn it over to Vivian.
Sweet, thank you, Stefan. I just want to double check. Y'all can't see my presenter view, and you can see my pointer, right?
Yes, and we can see your the words.
Yeah. All right, I had some problem with that earlier, so perfect. If you want to just stick my little face up here, that would be wonderful. Let's get started. So I'm gonna quick talk about some how we view antioxidants in our society right now. So a lot of food and drink commercials will use antioxidants as sort of a selling point for their products, but few of them actually give any explanation of what antioxidants even do or why they're good for our bodies, right? So this is a commercial by Palm, and they have this dragon in it that pops up, and it's supposed to represent the power of the antioxidants in their drink. And there's another one by Bai, which has John Legend singing in it, and he's just singing about how great antioxidants are. But at no point does he mention anything about how they might actually be beneficial to you. And we kind of link antioxidants with healthy diets and power fruits like blueberries and pomegranates, because that's like places we can find it every day. But so antioxidants work by removing free radicals from your body, and free radicals are super reactive compounds that just can interact with your DNA, your proteins, just stuff you don't want them interacting with. So antioxidants overall do help your body, but antioxidants, like most things, can harm your body in large doses, which can cause toxic pro-oxidant activity. Additionally, diets high in antioxidants are correlated with healthier lifestyles, healthier people, but taking dietary supplements containing antioxidants has no correlation to that at all, which is kind of an interesting fact. There's also some evidence that antioxidants could prevent tumor growth. And so some common antioxidants in your diet or that you might have heard of. So there's vitamin C, which is this lovely molecule right here. And these little black dots represent carbon atoms. The red ones represent oxygen atoms, and these little white ones represent hydrogen atoms. And then also we've got in the glutathione over here, we have blue, which is nitrogen, and yellow, which is sulfur. There's also vitamin E, vitamin A, selenium, carotenoids like beta carotene, glutathione again, selenium, and manganese. And these interact with radicals in your body, and the most prevalent type of radical in your body is reactive oxygen species, or ROS. So these are just like some basic examples. Surprisingly enough, they are made with oxygen and are highly reactive, if you couldn't get from the name. And hydrogen peroxide is kind of the hydroxyl radical. Basically, actually, like all of these are pretty big in your body. These two are really big. So what are reactive oxygen species? How are they formed? Why does oxygen do this while other atoms don't do this, or don't form these radicals quite as much? These are formed by incomplete reduction of molecular oxygen. And oxygen tends to accept one electron, right? So we've got, this is just normal ground state oxygen. It has 2 electrons in its 2p orbital here. It's pi star. So this is a non-bonding orbital, but you add an electron, the spin is important, right? So if you're adding 2 electrons to this, both of the spins have to be correct and matching with the 2 electrons this oxygen already has. to form a peroxide ion, right? If you're just adding one, only one of those electrons has to be. And another thing to point out is in the singlet oxygen, right? You've got one of these electrons has moved over with the other. And that's kind of like if you took the negative ends of batteries and tried to force them together, or the positive ends, right? Electrons repel each other. So this right here is a lot less energy preferred than just ground state oxygen, right? This isn't going to last for very long. And so those molecules are a little unstable, and they kind of either give away or take an electron. And so these are all, this is kind of a lot on the slide, but these are all different types of chemotherapy, right? These blue and orange words. These are all different kind of basic functions. So you've got, this is antioxidants, right? These little T's mean that it's inhibiting the antioxidants. So it's like lowering the amount of antioxidants in your body, right? And these antioxidants would normally inhibit reactive oxygen species, but these cancer therapies are actually lowering this level, which will raise this level, right? So this is all raising the reactive oxygen species level in your body. And then this is like basic metabolism functions that your body does that just naturally forms reactive oxygen species. And all of these cancer therapies are just kind of targeting these different functions, right? So, and kind of, if you can see this on a graph, we've got, this is like your normal cancer cells level of reactive oxygen species. And then you add on more, and suddenly it's too much for the cell to handle, right? It's reacting with proteins and DNA that it shouldn't be, and this is going to cause the cell to die. And this is kind of the idea behind some chemotherapies, not all of them, but some of them basically just work with this idea. So kind of to reiterate, right, you have chemotherapy and your cancer tumors both forming reactive oxygen species, and those reactive oxygen species are going to actually attack those cells and cause them to die. And so a lot of the side effects you get from chemotherapy are also due from this increase in reactive oxygen species in your normal cells, which is kind of an interesting point. So then you've got chemotherapy forming your reactive oxygen species again, but you're gonna, if you're taking antioxidants, and this is a fear that a lot of people have, it's gonna inhibit your reactive oxygen species, right? And then your cancer cells aren't gonna die, and they're just gonna keep proliferating. And this is kind of like a big fear and a big worry for a lot of people who are doing these tests and studies that we're going to look at later. Yeah, so you can see that instead of reaching up to this level like it should be, it's staying down here, or maybe even getting knocked down a bit if you add antioxidants, is what people are worried about. And so another pathway that a lot of people are hoping is what happens is that antioxidants aren't going to interact with your reactive oxygen species. They're going to work in a different pathway to sort of affect these cancer cells and cause them to die anyway. There's some evidence for both of these pathways, but for the most part, most of the evidence is pointing toward this pathway or this next pathway. So another reason you might want to put antioxidants in your body with chemotherapy is that chemo drugs can cause cancer cells to go into metastasis, right? So when a cancer cell metastasizes, it it slows down, right? It stops, it slows down its metabolism, it stops proliferating as much, and those are the things that that chemotherapy targets, right? So this increase in reactive oxygen species, the cell's going, oh no, I have too much ROS, I've got to slow down my metabolism. And this leads to drug resistance, right? Because now those drugs can't really distinguish it from your normal cell. And the idea is that if you add antioxidants back in, it'll go back into this weird cancer cell cycle, and your chemotherapy drugs then have a chance to kill them again. And the idea that, so reactive oxygen species could cause your normal cells to die or cause side effects of them, like a lot of people who take chemotherapy experience. But if you add dietary antioxidants in, those reactive oxygen species, that level is going to lower back to normaler levels for your cell that your cell can handle, and then you're just going to continue on your normal happy cycle for your normal cells. Radiation therapy is something we need to talk about really quick. So radiation therapy works by X-rays basically damaging your DNA. But while they do this, they also ionize water and oxygen, and that forms reactive oxygen species. And then those reactive oxygen species also damage your DNA. And this is important because chemotherapy and radiation therapy both use reactive oxygen species, while other cancer therapies aren't so heavily involved in reactive oxygen species. So they're going to be important to our pathways. So we've got this question, right? We have antioxidants that prevent tumor growth. We have chemotherapy, which kills cancer cells and prevents tumor growth, but we don't know how they interact. And kind of to, this image is a little too simplified for us, right? So we're going to move on to this one. So we have chemotherapy forming reactive oxygen species, and also generally inhibiting tumors in its own separate way as well. And then we have these reactive oxygen species and antioxidants inhibiting tumor growth. But on top of that, antioxidants inhibit reactive oxygen species, and tumors form reactive oxygen species. And if you're confused, I'm slightly too, because this is just a massive mess of things where it's like one thing helps something and then hurts something else that's trying to do the same thing as it. And this is kind of a problem that a lot of scientists are having, right? This isn't a very easy or straightforward sort of interaction between antioxidants and chemotherapies. So in vitro studies, there are studies performed on cells in the lab. These cells are generally grown on plates like this. And the example we're about to look into is where people tested the analogs of vitamin A and photodynamic therapies, which are just basically the compounds only start working once you expose them to light. So it's just a way of controlling the timing. And they used rat tumor cells. So this is sort of the viability of cancer cells when you use chemotherapy and antioxidants on them. So this is the percent viability of the cell. We want this to be low because they are tumor cells, so we want them to die. And this is our control, right? It's almost 100%, 200%. These error bars are like, it could be 100% viability, right? Once you add in this cancer chemotherapy drug, it lowers significantly. And same here with DOX, right? This is another chemotherapy drug. But once you start adding in these different vitamin A analogs, right, we don't really see a huge drop. Actually, this is the only combination that has a significant drop in comparison to just the plain old chemotherapy. And we see this a lot in a lot of these experiments where there will be a decrease that's noticeable to the eye, but not statistically significant. And it's very consistent throughout. But then if we go to clinical trials, which are trials based on human reactions to a drug, we'll see something a little different, right? So we're going to look at antioxidant supplementation and breast cancer prognosis in women undergoing chemotherapy and radiation therapy. And this trial studied about 2,200 participants, which is pretty big for one of these trials, right? And so we've got, this is the number of subjects, right? This is for these bars. And this is the recurrence of, the recurrence or death after 6 months of completing chemotherapy. And that's this line, this is the percentage, right? So for example, this is people who just did chemotherapy This is the total number of people who did just chemotherapy, and this is the deaths or recurrence or new cancer within 6 months out of that population, right? And this is people who took antioxidants and did chemotherapy. And we can see that their rate of recurrence or death greatly increased, right? This is like at 15%, and this is at like 20%, right? So that's like up a full 5%. And the same with radiation therapy and antioxidant. People who use antioxidants with radiation therapy, and people who took radiation and chemotherapy, and people who took antioxidants with that particular treatment plan, right? The thing that's interesting is that none of these numbers are statistically significant, right? Again, there's a trend, and you can see it clearly throughout quite a few studies, and there are actually like there's a lot more variance in what different studies find. But very few studies actually find much that's statistically significant. And we're just going to real quick go into why that might be, right? So there's some design problems that are, for the most part, out of the hands of the people who are making these experiments, right? So it's really hard to control variables in these, right? So you have people taking different types of chemotherapies. You have people taking different combinational therapies, right? When you start therapy to cure your cancer, you're generally not just doing one, right? You're probably doing different types of chemotherapies. You might be doing some radiation therapy. You might be getting some surgery. You very rarely do one thing. People will have different diets, and as we mentioned before, eating antioxidants in your diet seems to have more effect than taking a supplement does. There's also people taking different antioxidants, right? So different antioxidants could affect your chemotherapies in different ways, same as how different chemotherapies could be affected by antioxidants in different ways. There's the time that you actually get your chemotherapy done, and then when you actually start taking those antioxidants, that lapse between can really affect how those antioxidants are going to affect your treatment, right? If you give the chemotherapy, an hour to sit in your body before you take that antioxidant versus, a week, that's going to make a huge difference as to how much that antioxidant is actually affecting the therapy you're taking, right? There's small sample sizes, right? Very few of the studies I looked at had more than 20 participants. And it's really hard to get something that's statistically significant with such a small number of people, especially when you have so many different variables you need to account for within it. Another thing is homogeneous samples. So a lot of the studies done in different countries are like, oh, well, we tested all of the people in this small town. And it's still very valuable information, but again, it's just a very specific set of people. People have different types of cancer. They're treated different ways. And also antioxidants are probably going to affect them slightly differently depending upon the type of cancer. And then the wealth of individuals is also an important thing to pay attention to, at least in America, because our healthcare system is money-based, and if you have more money, you're probably going to be getting better treatment, right? So we want to make sure that it's actually based on the antioxidants you're taking and not based on something else like the amount of money you have or maybe race or something like that. So in the future, we need to make new trials that account for more of these variables, and a consensus between people working in the field as to how trial data should be collected and measured is also going to allow for comparisons between different trials, which will be helpful to figure out whether or not antioxidants negatively or positively affect chemotherapy. And that is the end. Any questions? If you have one, please just shout it out because I can't see everyone.
I got you, Vivian. I'm happy to moderate the questions.
Sweet. Sounds
I'll go
great.
ahead and call on anyone with the questions.
Okay.
Thank you, Vivian. Hooray. Do we have any questions for Vivian?
I have a quick question.
Yes.
Sure.
So what are some of the ways that you think a good clinical trial would go to test some of these different parameters?
Yeah, so the harder, I can go back to it. So the easy ones to do, right, are paying attention to like the type of antioxidant, the type of chemotherapy, and other therapies, and like the type of cancer people have. The ones that are a bit harder is like diet, right? So I would definitely be, I've seen a lot of ones where it's like they follow up with people 2 years after the trial and ask, oh, well, what were you eating before you started? What were you eating when you were in the trial, in doing chemotherapy? And what did you eat right after? And it's like, well, I'm not going to remember what I was eating 4 years ago, let alone 2 years or 2 years ago or 3 years ago, right? And that's kind of what they're asking. So I think I would definitely, if I were running this, ask like, you know, what are you eating now? Like have a questionnaire at the beginning, the middle, and the end of someone's treatment. And you know, the money thing, a lot of people did a pretty good job of considering, you know, your race, money, where you're living, stuff like that. That's just like kind of basic stuff that surprisingly I didn't find in all of them. So I think I would definitely make sure that's in there. And then the hardest part, I think, for people is getting a good sample size, right? Because even in the study that had 2,000 people, you know, probably like, let's say half of them took antioxidants. From the graph above, you can see way less than half did, right? But if half of them were doing antioxidants, we had like 1,000 people, right? Not all of those people are taking the same therapy. And you can't really compare between different therapies. And then not all of them are taking the same antioxidant. And you can't really compare between antioxidants, right? The reason I made the graph above is because it's not really like you can't exactly compare like that. It was just to kind of help because otherwise I just have a bunch of numbers on the screen. But so that's kind of important, right? And the reason that none of those numbers were statistically significant is because you probably have 5 people taking a specific antioxidant out of those 2,000 that are also taking the same chemotherapy and the same other therapies together, right? Even if you have a massive group like that. So I think the biggest thing would be actually to try to make like a massive database where you just have enough variables inputted in it that you can actually start comparing between people, because there's no way to do it in just one study. Like it won't happen in one. I hope that answers your question.
Yeah, I love the
I went on a
database
tangent.
idea.
All right, Camille, it looks like has a question. Go ahead, Camille.
Sweet.
All right. First off, congrats. That was a great talk. So my question is basically, you probably did a bunch of reading for this. And I was just wondering if there were any particular like antioxidants that popped up that seemed to be like studied a lot more than like anything else.
Yeah, so vitamin C is a huge one that a lot of people studied. And it was kind of funny because people either found that it really didn't work or it really did. Like it was one of the ones where it was consistently, they were like, this is the one we found was significantly like reduce the amount of chemotherapy you had to take, like reduce the side effects, and like just completely wiped out the cancer in people. And then the next paper I'd read would be like, vitamin C sucked. It like really increased the death rate of people. It was awful. So that was a really funny thing to see. Like everyone had very different opinions on vitamin C, and they were very polarized kind of opinions. I shouldn't say opinions, it's just data, but.
Cool. Thanks.
Vivian, I'm really interested in the different mechanisms of the different chemotherapeutics, and then how you showed that their kind of relationship to reactive oxygen species and thus antioxidants would be really different. In the studies you looked at, are they trying to take that into account? I know you said that
a little bit, yeah.
a lot of them have different chemotherapeutics that they're taking and everything.
That's not the right slide. Yeah, it's kind of, oh, you're all hidden. I tried to expand it so I could see more, and then I couldn't find the slide. Yeah, so a lot of the papers are very odd because it's like you'll get the paper and they'll give you all of these things where they're like, well, we found these things that are significant, right? Like if you had if you took some kind of alkylating agent and you had vitamin C, there was a blank % chance that you would die, you know, and this was statistically significant. But then when they give you the actual numbers, it's all grouped together as like people taking chemotherapy, people taking radiation therapy, people who took like sometimes the type of antioxidant is spelled out. Sometimes it's just people who took antioxidants, people who didn't. So it's kind of weird because there's no way to check like the actual numbers they give you. And they don't like put in graphs or anything. It's just like, this is the one that we want to tell you about, which is wild. And I'm definitely not answering your question. Can you repeat your question?
No, I mean, that really is, I was kind of hoping you would talk about the methodologies that they're doing in these studies, because it seems to me like not trying to zero in on a single chemotherapeutic is a huge challenge to getting any kind of statistical reading out of this.
I mean, I think the biggest problem is that, you know, even in the example I gave where they had 2,000 participants, right? That was one of the largest ones that I could find. And even there, it's like the numbers just aren't enough for the number of people taking different antioxidants and different types of therapies, different combinations of therapies, you know, which is why I really think that we need to try to get some kind of database where you can kind of compare different trials or different participants from trials in there. But that's also very tricky because you have to be able to measure all of those different variables in the same way then. And there hasn't been any consensus on how to measure a lot of them in the community.
Thanks. Further questions for Vivian? Okay, going once, twice, sold.
Yeah.
Thank you, Vivian, for a wonderful talk. Please join me in thanking you one more time. Maybe unmute your video if you feel up to it. Say congratulations. All right.
Yeah, I can stop sharing.
All right. So thank you, everybody, for joining us. Thank you, Vivian, for the excellent talk. And we'll hopefully see many of you next week for Clayton's talk on Monday. I want to make sure I had the right date. So Clayton will be speaking to us on Monday about blood-sucking bugs and the war between oxidative stress and hemozoan crystals. So the hit parade just keeps on. Thanks, Vivian. Great job.